If you want to live a long time, you cannot ignore the kidney. It is not a background organ. When kidney tubules start to senesce, when they stop dividing, stay metabolically active, and leak inflammatory signals, the whole body feels it. Blood pressure control worsens, cardiovascular risk rises, waste clearance falls, bone and anemia problems follow. In diabetic kidney disease, that tubular aging is not a late side effect. The paper we are discussing today treats it as a central driver of the disease.
The paper is from Liu, Zhang, Zhao, He, Lang, and Sun, published in the FASEB Journal in 2026. Title: SGLT2 inhibition attenuates renal tubular senescence by suppressing CTRP1 mediated glucotoxic stress in diabetic kidney disease.
What they add is a specific upstream mechanism. Not just high sugar is bad for tubules, they argue that a protein called CTRP1 turns up SGLT2, the main glucose reuptake pump in the proximal tubule. That increases glucose entry into the cell. That glucotoxic stress pushes the tubule towards senescence. And when they blocked SGLT2 with a low dose of dapagliflozin, that axis quieted down and the senescence phenotype eased.
So, this is both a diabetes paper and an aging paper.
Yes, diabetes is the stressor they used. Senescence is the aging process they measured. The health question is whether interrupting that glucose uptake loop is one reason SGLT2 inhibitors protect kidneys, and whether that matters for longevity.
Start with the clinical problem. Why should someone who cares about living longer care about renal tubules?
Because the kidney ages early and loudly. After midlife, glomerular filtration rate declines in most people. In diabetes, that decline accelerates. About 30 to 40% of people with diabetes develop diabetic kidney disease, and DKD is still a leading cause of kidney failure worldwide. Kidney failure is not only a dialysis problem, it is a systemic aging problem. Higher cardiovascular death, frailty, infection risk, and loss of independence. For years, the field focused on the glomerulus, the filter. That is important. But tubules do most of the energy intensive work. The proximal tubule reabsorbs the bulk of filtered glucose and sodium. In hyperglycemia, it is forced to handle an enormous solute load. That costs ATP, generates reactive oxygen species, damages DNA, and can lock the cell into senescence. A senescent tubular cell is not resting, it is arrested. High P16, high P21, and it secretes a SASP, cytokines, chemokines, matrix remodeling enzymes. Those signals recruit inflammation and activate fibroblasts. Fibrosis follows, function falls. So senescence is both a result of injury and a cause of further injury. That is why this paper calls tubular senescence a defining pathological feature of DKD and a key driver of progression.
So, if you only lower blood sugar in the bloodstream, you might still leave the tubule overworked.
That is the key distinction. Systemic glucose and intracellular glucose flux are related, but they are not identical. SGLT2 sits on the apical membrane of the proximal tubule and pulls glucose back into the cell. If that transporter is upregulated, the cell can take in more glucose than is healthy even before you consider the rest of the body. The authors went looking for what sits upstream of that transporter.
Walk us through what they actually did. Human data first.
First, association. They looked at CTRP1 in diabetic kidney disease of increasing severity. CTRP1 is C1q TNF related protein 1, an adiponectin family secreted protein. It is not a household name, but it sits at the intersection of metabolism, inflammation, and tissue signaling. In their data, CTRP1 expression rose as DKD got worse. That already suggests it is not a passive bystander. Then they correlated CTRP1 with canonical senescence markers. CTRP1 tracked with P21 at R equals 0.498 and with P16 at R equals 0.494. Those are moderate correlations, not destiny, but they are consistent in direction. More CTRP1, more of the molecular signature of cellular aging in the kidney.
How did they get from correlation to mechanism?
Loss of function and gain of function. On the loss side, they examined data from CTRP1 deficient mice. SGLT2 was consistently downregulated when CTRP1 was missing. That is an important clue. CTRP1 is not just nearby damaged kidneys. Removing it appears to take SGLT2 down with it. On the gain side, they used NRK52E cells, a standard rat proximal tubule epithelial line. When they overexpressed CTRP1, SGLT2 expression rose and glucose uptake rose. The effect was stronger under hyperglycemic conditions. That matters. It means the axis is stress amplified. In a high glucose environment, CTRP1 does not just exist, it appears to worsen the cell's decision to pull even more glucose inside. So the working model is the DKD milieu raises CTRP1. CTRP1 raises SGLT2. SGLT2 raises tubular glucose uptake. Glucotoxic stress accelerates senescence.
In the animal experiment, the part listeners will ask about.
They used a fairly aggressive rat model, high-fat diet, streptozotocin, and unine nephrectomy. High-fat diet plus a pancreatic toxin plus removal of one kidney. That combination produces metabolic disease plus reduced renal reserve, which is closer to human progressive DKD than a single mild insult. They treated with dapagliflozin at 0.1 milligrams per kilogram per day. That is a low dose. I want listeners to hear that. They were not blasting the system with an enormous pharmacologic hammer. Even at that dose, metabolic abnormalities improved, kidney function improved, and tubular senescence was attenuated. Mechanistically, dapagliflozin suppressed activation of the CTRP1-SGLT2 axis, reduced glucose uptake in tubular cells, and markedly decreased the proportion of tubules that were double positive for both CTRP1 and SGLT2. That last point is conceptually important. It is not only that average expression of each protein fell, the cells co-expressing both pieces of the machine became less common.
So the paper's claim is not SGLT2 inhibitors help kidneys, full stop. We already knew that from outcome trials. The claim is how, at least in part.
Correct. The authors say this is a previously unrecognized CTRP1 SGLT2 signaling axis linked to glucose mediated tubular senescence, and that it helps explain SGLT2 inhibitor renoprotection. I think that is the right level of claim. They did not prove this is the only pathway. They showed it is a coherent, experimentally supported one.
Translate that into health span. If this axis is real, what changes for a person trying to live longer?
Three things. First, it strengthens an idea longevity research sometimes underweights. Organ specific metabolic load is an aging input. In the kidney, the load is glucose and sodium reabsorption. Reduce the load, and you may reduce a senescence trigger. That is local geroscience. You are not curing aging. You are lowering a chronic injury signal in an organ that limits both lifespan and quality of life. Second, it fits a larger SGLT2 inhibitor story this field already watches. These drugs began as diabetes medicines. Large trials then showed they reduce kidney failure, heart failure hospitalizations, and in many analyses, death, including in some people without diabetes. Separately, aging biology papers have shown that SGLT2 inhibition can lower kidney senescence markers, reduce oxidative stress, engage ketone body and NRF2 antioxidant programs, and in some models reduce senescent cell burden more broadly. Canagliflozin has even extended lifespan in certain mouse studies, including work where immune surveillance of senescent cells was part of the story. This paper does not repeat those experiments. It adds a missing gear, an upstream protein that may set how much SGLT2 the tubule deploys, and therefore how hard the cell works under sugar stress. Third, the health implications are systemic even though the mechanism is tubular. Preserve tubules and you are more likely to preserve filtration, volume control, and the kidney's endocrine jobs. That affects heart failure risk, blood pressure, anemia, mineral bone disease, and the chronic inflammation aging researchers call inflammaging. A kidney that is less senescent is a body that is less inflamed.
The question people will screenshot: should healthy people take dapagliflozin to live longer?
No, this paper does not support that. The model is diabetic kidney disease, not normal aging. The human data in the paper are associational. The causal work is in cells and rats. CTRP1 itself is also biologically messy. In other contexts, it has been described as protective against fibrosis or associated with slower CKD progression. So we should not turn CTRP1 into a cartoon villain. Context matters. Diabetes versus obstruction versus obesity versus ordinary aging may not use the protein the same way. SGLT2 inhibitors also have real harms: genital infections, volume depletion, rare euglycemic ketoacidosis. They are not appropriate for everyone. Longevity enthusiasm is not a prescription. What is justified is this: if a clinician already has a reason to use an SGLT2 inhibitor, type 2 diabetes at high risk, heart failure, or chronic kidney disease, this paper adds a mechanistic reason to believe part of the benefit may be anti-senescent in the tubule, not only hemodynamic or glycemic. That is medically relevant. It is not a license for off-label anti-aging use.
And for people without those diagnoses?
The durable lesson is still useful. Protect the kidney as a longevity organ. Blood pressure control, avoid unnecessary nephrotoxins. Treat diabetes early if it appears. Stay out of obesity when possible. Respect that high filtered glucose is not free. The tubule pays for it. This paper names one of the molecular cashiers, CTRP1, working through SGLT2.
Before we close, 3 listener style questions. 1. Is CTRP1 a new longevity target?
It is a candidate node, not a drug target yet. Nobody in this paper inhibited CTRP1 directly in the rats. They inhibited SGLT2 and watched CTRP1 SGLT2 signaling fall. If someone designs a safe CTRP1 modulator, this work would justify measuring tubular senescence. That is the next experiment, not the current conclusion.
2. If I already take an SGLT2 inhibitor, is my kidney aging more slowly?
We cannot say that about an individual listener. We can say the class protects kidney outcomes in large trials, and this paper offers one cellular aging explanation in a diabetic model. Your own trajectory still depends on blood pressure, albuminuria, glucose, heart function, and time.
3. What should I watch for in follow up research?
3 receipts. 1. Human tissue or good biomarkers showing CTRP1 and SGLT2 moving together with P16 and P21. 2. Whether dapagliflozin or another SGLT2 inhibitor lowers those markers in patients, not only in rats. 3. Whether the same axis exists in non-diabetic kidney aging. Until that third receipt arrives, this remains a strong DKD mechanism with a plausible geroscience implication, not a completed longevity therapy.
4 things to keep.
1. In diabetic kidney disease, proximal tubule senescence is not cosmetic. It helps drive the disease. 2. CTRP1 rises with disease severity and correlates with P16 and P21. Losing CTRP1 is linked to lower SGLT2. Adding CTRP1 in tubular cells raises SGLT2 and glucose uptake, especially in high glucose. 3. In a high-fat, streptozotocin uninephrectomy rat model, low-dose dapagliflozin improved metabolism and kidney function, reduced tubular senescence, suppressed the CTRP1 SGLT2 axis, and cut the number of double-positive tubular cells. 4. For longevity, this is evidence that a widely used cardiorenal drug class can interrupt a glucotoxic aging pathway inside the kidney. It is a mechanism paper, not a lifespan trial. The honest position is interest plus restraint.
That was Longevity and Live Forever. Paper: Gu and colleagues, the FASEB Journal, 2026.
If you have diabetes, heart failure, or kidney disease, decisions about these drugs belong with your physician. The science is encouraging. It is not a substitute for clinical judgment.
Living longer is often less about a miracle molecule and more about keeping workhorse tissues from becoming senescent too soon.
The kidney is one of those tissues. Thanks for listening.